Hello and welcome to HN 501 Human Development and Human Behavior in Context. I'm Dr. Lisa Marie Jones and I'll be your professor for this course. This course will provide you an overview of human development, emphasizing the application of theory and knowledge to methods of intervention that promote healthy functioning and autonomy. I'm recording this supplemental lecture for our first week of course because we spend a great part of Unit 1 seminar discussing course expectations, which doesn't usually allow enough time for us to cover the unit topic. I'll also provide a PDF of the slide handouts in the announcement section of this course. This lecture will focus on brain development and what's called epigenesis. So, both heredity factors and environmental factors shape human development. Genes can't do anything without environmental input, and the environmental effects are shaped by genetic constraints. As a human service practitioner, whether you're a medical or behavioral health care provider, an administrator working with employees, or a teacher working with students, it's important to understand this relationship between genes and environment. Knowledge of the relationship between nature and nurture is going to improve your own ability to provide specific therapeutic interventions and realistic goals. And it'll also allow you to help others to understand the complex interplay of hereditary factors and environment on physical and behavioral outcomes. Because they carry the same biological inheritance, we oftentimes will look at identical twins to help us understand more about the impact of heredity and environment on development. With twins, we see that neither hereditary factors or environmental factors work alone. Cell specialization shows us that hereditary factors are not solely in charge of development. The term epigenesis was originally used to describe the development of specialized cells and systems of cells, like the nervous system or the digestive system, from an undifferentiated zygote. The term described genetically similar things and how they can have different outcomes, which was a mystery. The term epigenesis has evolved with the progression of science, and it's now defined more specifically as a set of processes by which factors outside of hereditary material itself can influence how hereditary material functions. These factors that we're talking about are environmental. This can include the chemicals in the cytoplasm of the cell, which constitutes the immediate environment surrounding the chromosomes, to factors in the cells and tissues adjacent to the cell, all the way to factors beyond the body itself, such as heat, light, and even social interaction. Okay? But as you will learn, the chemicals in the cytoplasm of the cell are themselves partly determined by hereditary material in the chromosome, okay, in the chromosomes. These chemicals can move beyond a cell to influence adjacent cells and ultimately influence behavior in the outside environment. From the beginning, inherited stuff and the environment are engaged in an intricate process called coaction, so that neither one ever causes any outcome on its own. The structural and functional properties of the hereditary material and the environmental factors have reciprocal effects, meaning that they both influence each other. Okay, so when the question is asked, is it nature or nurture, it's both. You probably already know that inheritance of traits begins with reproduction right at conception, when a man's sperm fertilizes a woman's egg or ovum. The human ovum is a giant cell with a nucleus that usually contains 23 chromosomes, which are the physical structures that are the vehicles of inheritance from the mother. The sperm, in contrast, is a tiny cell, but it also usually carries 23 chromosomes, which are the father's contribution to inheritance. The ovum's nucleus is surrounded by cellular material called cytoplasm. During fertilization, the sperm penetrates the outer membrane of the ovum and makes a relatively long journey through the ovum's cytoplasm to finally penetrate the nucleus, where the sperm's outer structure disintegrates. The sperm's chromosomes become part of the nuclear material in the fertilized ovum, which is now called a zygote. The zygote usually contains 46 chromosomes, or 23 pairs of chromosomes. One member of each pair came from the mother, or the ovum, and one from the father, or the sperm. 22 of these pairs are matched, and they're called autosomes. In autosome pairs, the chromosomes look and function alike, so that's why they're called autosomes. The chromosomes of the 23rd pair are called the sex chromosomes because they have an important role to play in sex determination. In female zygotes, usually the 23rd pair consists of 2 matched chromosomes called X chromosomes, and you see there in that picture what an X chromosome looks like. But the male zygotes have a mismatched pair. They have an X, typically, I'll say, they have an X chromosome from their mom, but a much smaller Y chromosome from their father. The cells then divide and continue to divide, which is called mitosis. After about 2 weeks, the mass of duplicated cells of the zygote migrates down the mother's fallopian tube into the uterine lining and implants itself by attaching itself to that uterine lining in order to grow and develop. The zygote is now called an embryo. So let me talk to you a little bit about sex differentiation. And as I was saying earlier, that the female usually is made up of XX chromosomes, so it would be 2 of those large X chromosomes there, and the male is typically a mismatch pair, which would be XY. And the reason why I said typically is because sex differentiation is a process of development of differences between males and females from the undifferentiated zygote. And as individuals develop from zygotes into fetuses, into infants, children, adolescents, and eventually into adults, sex dichotomous differences are not absolute. There's many individuals who are exceptions to the typical XX female and XY male. Sex differences might be induced by specific genes, or by hormones, by anatomy, by social learning. Some of the differences are entirely physical, for example, a presence of a uterus, and some differences are just obviously are of social learning and customs, and usually we call that gender or gender identity, like hair length, those kind of things. Many differences like gender identity we find are influenced both by biological and social factors, so we do see the role of nature and nurture. But for now, we're just going to discuss the physical differences that can occur. Okay, so the first weeks of gestation, the first 8 weeks of gestation, we're all female. That's right. It's the default sex. So phenotypically, what we look like on the outside and even what we look like on the inside are more feminized, okay? We're made up of all the same stuff, whether you're male or female. We used to think that the presence of the Y chromosome made a person a male and its absence made a person a female, but we now know that that's not true, and we'll discuss that in a little bit. So we're all female, and we all contain what's called the Mullerian ducts and the Wolffian ducts, okay? And then you have these gonadal streaks or bipotential gonads. And then you have the cloaca, okay? The cloaca is eventually going to turn into the bladder, urethra. We'll talk a little bit about what causes the masculinization in a minute. But if the child masculinizes, on the inside, the Mullerian ducts degrade. Then the Wolffian ducts become the epididymis and ductus and vas deferens, okay? And then you see how those gonads become testes, and then the urinary bladder and urethra from the cloaca. And if the child stays feminized, the Wolffian ducts degrade, and the Mullerian ducts become uterine tubes and uterus. Okay, let me switch this. You have parents, and most women are 46XX and most men are 46XY. So a mother can only give an X chromosome. A father can give an X or a Y chromosome. And like I said earlier, we once thought that the presence of the Y chromosome made a person male or female. But we have recently found out that really it is this SRY gene that is usually located on the Y chromosome, but it can translocate to the X chromosome that masculinizes a fetus, typically. But it's not the only thing that can masculinize the fetus. Okay, and so we'll talk about those differences. You can have what's called chromosomal sex determination and hormonal sex determination. Through accidents of chromosomal sorting, during what's called meiosis, during the sperm and egg production, or there are some people who inherit an XXY combination, but would still be considered male with Klinefelter syndrome. Others inherit only an X, and thus denoted XO. Sometimes you'll see 45X. And they're usually female. Not only females who are XX and males who are XY. There's a wide range of chromosome complements. And like I said, hormonal imbalances and phenotypic variations will also determine the sex. So you can have this SRY gene that translocates, and so you have this father that gives an X chromosome, but that X chromosome has SRY gene on it. So that child ends up being male. Because remember, at that 8th week of gestation, that child starts to masculinize if that SRY gene kicks in and says, masculinize this baby. Then you have those, the labia that would be open, the opening of that 8-week gestational age baby would close, and it would be, that's where the gonads would end up, where the testes would go. And if the child stays feminized, the gonads will stay up in the abdominal area and the opening will stay open. If you look at a little boy when they come out, it looks like their scrotal sac has been sutured, and that's because it was once open and it comes together there. Okay, and then they start to masculinize. We do have what's called XY-AIS. This child can either be male or female, and what happens is that SRY gene on that Y chromosome doesn't tell the androgens to kick in. So there's some kind of mutation sometimes, or the androgens just don't kick in. Okay, you can also have an XX male who does not have an SRY gene, but was exposed to androgens in vitro. This child can also be female. Sometimes these children have ambiguous genitalia when they are born with intersex conditions, and it's not so readily visible. So what would have been a clitoris with that 8-week-old child, or I'm sorry, 8-week-old gestational child, becomes either that it becomes a penis. So we all have that outer phenotype that is a phallus that will remain more feminine if there is not an exposure to androgens, or it will become more penile-like. as it's exposed to androgens. Now, like I said, you can have variations of that to where a child has ambiguous genitalia, where maybe they might still have an opening, but they have an enlarged clitoris or a micropenis. They can have what's called hypospadias, where they have a penis and a closure, but their opening, their urethra or opening is, and the ureters and the urethra opening is down lower on the penis instead of at the tip. So there's just differences that you can see phenotypically. You also have trisomy X, so that would be a female, but you also have XYY, and like I said earlier, XXY, that's Klinefelter syndrome. A lot of times these people will end up being not able to reproduce, but not always, okay? You have XYY, which used to be referred to as the super male. And then you also have what's called polysomies, where you have more than, that would be like the XXX or XYY or XXY, and then you have pentasomies, so it can go all the way up to 5, 6 sex chromosomes. We see so the 47 would be trisomies, okay? So there's a wide variation that we see. And then there's also what's called mosaicism, meaning that the person has 2 or more cell lines with different genetic signatures, and this person can be XX, XY, so 46XX and 46XY. They can be 46XY, 47XXY. They can be 45X, 47XXX. So the theory behind that is that the child was going to be a twin, male or female, with the XXXY, or twins with, just a different sex chromosome makeup. And then during that mitosis period, the cells came back together and they were fused together, and that person is mosaic. But we don't know for sure if that's the cause. When we say gender, we're typically describing terms of masculinity and femininity. It's a social construct that varies across different cultures. There's a number of cultures that with greater diversity in gender, but sex, usually when we're talking about sex, we're talking about biological, okay? So there's so many different variations, and there's so many different things that can cause you to be male or female, or somewhere in between, but there's usually a dominant gender that comes out. The child will tell you what they feel closest to. Okay, so let's move along. The chromosomes in the nucleus of the cell are made of a remarkable organic chemical called deoxyribonucleic acid, or DNA. Long strands of DNA are combined with proteins called histones, wrapped and compacted to make up the chromosomes that we can see under a microscope. Chromosomes can reproduce themselves because DNA has the extraordinary property of self-replication. Genes are functional units or sections of DNA, and they're often called coded sections of DNA. For each member of a pair of chromosomes, the number and location of genes are the same. So genes, like chromosomes, come in matched pairs, half from the mother, like we said earlier, and half from the father. So half from the ovum, half from the sperm. Genes provide a code that a cell is capable of reading and using to help construct a protein, a complex organic chemical made up of smaller molecules called amino acids. Proteins in many forms and combinations influence physical and psychological characteristics and processes by affecting cell processes. The DNA code is a long sequence of molecules of 4 basic chemicals called adenine, cytosine, guanine, and thymine, identified as ACGT. In a process called transcription, intertwined strands of DNA separate, and one of the strands acts as a template for the synthesis of a single strand of messenger ribonucleic acid, or mRNA. The code sequence is replicated in the mRNA. Different sections of the gene's code can be combined in different ways and result in different forms of RNA. The second step is called transcription, and this is when the cell reads the messenger RNA code and produces a protoprotein, a substance that can become a protein. So again, the cell can produce several protein variations from the same protoprotein. Different cell climates, combination of chemicals can encourage different protein outcomes. Epigenesis is one reason that identical twins can have the same genotype, so the full complement of an organism's genes, but not have identical phenotypes or the physical and behavioral traits. Okay? So their genotype is exactly the same because they come from a single zygote. Usually after a zygote divides for the first time, the 2 new cells stick together and continue the cell division process, leading to multicelled organisms. But in identical twinning, after the first cell division, the 2 new cells fail to adhere, and each of the new cells continues the cell division process separately. Each of the 2 clusters of the cells can develop into a complete organism, producing identical or monozygotic twins. Okay? Yet even though they have the same genotypes, their environments may diverge, even prenatally. So epigenetic effects mean even monozygotic twins, which are identical hereditarily, show different physical and behavioral characteristics. So their experiences cause changes in cellular processes that affect their genes' expressions. Okay? Typical development. So we see in typical prenatal development that there's this pretty amazing order and continuous progress from a single fertilized cell to a highly differentiated organism with so many different interconnected and efficiently functioning systems. The 9-month gestational period, or the 40 weeks that of the zygote, about 2 weeks from fertilization to implantation. The period of the embryo is from about the 3rd week to the 8th week. And this is that's when you start seeing the sex differentiation happen. When most of the body's organisms, systems, and structures are forming. And then finally, the period of the fetus is from the 9th week until the birth. And when the reproductive system forms, gains body weight happen, and the brain and the nervous system continue to develop pretty dramatically. Typical development depends on the genome to code for the products that the body needs to grow and function normally. And it depends on the environment to provide a normal range of inputs from nutrients and then also social interactions in order for the gene to gene expression to be properly timed and regulated. Okay. So we usually are going to be covering typical development, but there's always going to be a section in each chapter that discusses atypical development. So what role do genes and environment play in atypical development? We see deviations in either the genome or the environment that can push the developing organism off course. Okay, so when we look at hereditary diseases, they can occur as a function of a defective gene or a wrong number of chromosomes. Some disorders caused by dominant defective alleles don't kill their victims in childhood, but they can be passed on from one generation to another. When one parent has the disease, each child has a 50% chance of inheriting the dominant defective gene from the parent. Some of these disorders are mild in their effects, like farsightedness, for example. Others are more lethal, and sometimes they become lethal later in life. Among one of the most famous of those is Huntington's disease, which causes the nervous system to deteriorate, usually beginning between 30 and 40 years of age. And the symptoms can be pretty startling when you see them. They include uncontrolled movements, in increasingly disordered psychological functioning, eventually ending a person's life. In recent years, the gene responsible for Huntington's disease has been identified, okay? And then we have sickle cell anemia and progeria that are caused by defective gene alleles. And occasionally, a zygote will form containing too many chromosomes or too few, usually because of problems in the production of either the ovum or the sperm. Such zygotes don't often survive, and when they do, the individuals usually have multiple physical or mental neurological issues. One of the most common and well-known of these disorders is trisomy 21, which is also known as Down syndrome. So starting right at reproduction, right there at conception, the environment is an equal partner with the genes in the human development. And what constitutes the earliest environment beyond the cell is the mother's womb, right? But it's also everything outside of the womb as well. Every level of physical and social and cultural context. So if the mother is stressed by marital conflict, or if she just has this embedding of stress that she's lived with for her, throughout her life, that can affect the developing fetus or influence it, impact it. That's a biochemical change that's happening in the uterus when there is stress that's experienced, the increase in cortisol levels and epinephrine and such. Environmental substances and agents can cause harm to the developing fetus, and those are called teratogens. The name comes from the Greek, which literally means monstrosity making. Some teratogens, like alcohol, tobacco, and other drugs, are going to be coming into the body that way. But there's also things like infections, for example, HIV/AIDS. Then there's environmental hazards that the mother can be exposed to. These, like lead, for example, that's also a teratogen. Teratogens are problematic because they add something to the ordinary fetal environment, and they're intruding on the developing system, and they're driving it off course. So when there are contextual factors that belong in the ordinary fetal development that are missing or in short supply, this can also cause a problem. So, like if the child is not getting adequate nutrition, protein, essential vitamins, minerals during that prenatal or early postnatal development, the infant's physical, socio-emotional, and intellectual development can be compromised. And epigenetic alterations seem to be at the root of these developmental issues. Okay? So early prenatal development on the brain. So we have, after 2 weeks of gestation, cells from the embryo's upper surface that form a sheet that rearranges itself by turning inward and curling into a neural tube. This is called neurulation, excuse me. And it's the start of the fetal central nervous system's development. Around the 25th day of gestation, the neural tube makes a curved shape, and at the top of this C-shaped embryo, there's 3 distinct bulges that appear, which eventually become the hindbrain, the midbrain, and the forebrain. Okay? Cells from the interior surface of the neural tube reproduces to form neurons or nerve cells that will become the building blocks of the brain. Neurons increase at a rate of 1/4 of a million per minute for 40 weeks to create 100 billion cells or neurons rather, that make up a baby's brain at birth. About 1/2 of these neurons will be unnecessary, or they won't be used and will be destroyed later in life. Supporting cells called glial cells stretching from the inside of the neural tube to its outside, providing a type of scaffolding for your neurons, guiding them as they ventured out on their way to find their final destination. Okay. Those neurons that developed first migrated only a short distance from your neural tube and were destined to become the hindbrain. Those that developed later traveled a little further and ultimately formed the midbrain. Those that developed last migrated the farthest to populate the cerebral cortex of the forebrain. So in your hindbrain, you have the medulla, pons, the cerebellum, and reticular formation. That regulate autonomic functions. And then you have in the midbrain, the superior colliculi, the inferior colliculi, the substantia nigra, and that's involved in vision, hearing, and consciousness. And then you have in the forebrain the cerebrum, the thalamus, the hypothalamus, and limbic system, which handles neural communication. Okay? And here's the stages of the brain developing. So you see those 3 bulges. They're at the 25 days and what they become. Okay, so now we're going to talk a little bit about the structure and function of neurons. Neurons in your brain are one of nature's most fantastic accomplishments. Okay, so they come in various sizes and shapes. They're composed of a cell body with a long extension or axon that you'll see in this picture here. We'll come back to that. Okay. And that axon is kind of like a cable that's attached to a piece of electronic equipment. The axon can be quite long relative to the size of the cell and the other structures of the cell, because it needs to connect, or what we call wire, various parts of the brain. Like in the textbook talks about the visual thalamus to the visual cortex. Okay, at the end of the axon there are axon terminals. Okay, so you'll see that in this picture. You see those terminal endings. These terminals contain tiny sacs of chemical substances called neurotransmitters. And growing out from the cell body are smaller projections called dendrites, resembling little branches which receive messages or transmissions from other neurons. The unique structure of the neuron equips it to do its job efficiently when a neuron responds to some excitation or when it fires. You see this electrical impulse or message that travels down the axon to the axon terminals, and the sacs in the axon terminals containing the neurotransmitters burst and release their contents into the space between the neurons called the synaptic gap. About 100 different neurotransmitters have been identified, and many more are likely to be identified. Okay, so we're looking at not just the widely studied serotonin, acetylcholine, glutamate, gamma amino butyric acid, or GABA, that's impacted by alcohol. You see that. Or epinephrine, adrenaline, or norepinephrine, noradrenaline, or dopamine. There's many more besides that. Those are just the ones that are really common that people know about and that have been extensively studied. Some of them are more common in specific parts of the brain than others, and they're chemical messengers that stimulate the dendrites or the cell body or the axon of the neighboring neuron to either fire, this what we call excitation, or stop firing, what's called inhibition. If the message is to fire, then the speed of the resulting electrical impulse is increased when the glial cells wrap themselves around the axon, causing this facilitation conduction. This phenomenon is called myelination. And this begins prenatally for neurons in the sensory motor areas of the brain, but also happens later in other areas. This brain maturation process continues throughout much of the lifespan. The term white matter refers to the bundles of myelinated axons, and gray matter refers to the bundles of cell bodies, dendrites, and unmyelinated neurons. Okay, so those darker spaces do not have myelin, and the white spaces do, okay, when you're looking at the brain. So this is a picture here of what we were just talking about. And then you see those nodes of Ranvier in between the myelin sheath. You see how it wraps around. And then you can see the terminal endings at the end of that axon. And then those dendrites. And then the nucleus. So the myelin is going to speed up the communication. And you'll see later when we talk about the corpus callosum and the two hemispheres of the brain and how it speeds up that connection and communication and how the child becomes more coordinated as that develops. Okay, so development is progressive adaptation across multiple levels of functioning. Humans manage these adaptations with a stress response. Stress has been defined as a non-specific response to any demand. It can be positive, which is called eustress, or it can be negative, which is called distress. Organisms have a way of responding to threats, and it's referred to as the general adaptation syndrome. And it has three phases: the alarm phase, when the threat is first recognized, and then when the body prepares for fight or flight. The second is the resistant phase, during which the body's stress response is active and it continues to resist the effects of the stressor. And then you have the third and final stage, which is exhaustion, which occurs if the struggle persists to the point of complete resource depletion. We see depression, illness, or even death can occur after severe prolonged stress. People who are faced with chronic stress can have what's called allostatic load, and it can cause morbidities or health issues, and it can also cause mortality to be lower. When faced with acute stress, you have these physical symptoms that are less important for your survival, that are temporarily sidelined. So the resting and the digesting and reproduction don't happen when there's stress happening, okay? A more intricate explanation of the stress process is called allostasis, and it's composed, well, what happens is you have this central nervous system that controls multiple interacting regulatory processes. And so what it does is it maintains this balance through what's called adaptation. And allostasis allows for adjustments to be made within a range of possibilities across a variety of systems that suit the circumstances. And instead of returning to a fixed set point, it looks for the best balance for each specific challenge that it's found. Okay, it's called that's called allostatic accommodation. Your nervous system, your immune system, and your endocrine systems are the primary interconnected networks that facilitate this adaptation process. So it's usually, we call the stress management apparatus the hypothalamic pituitary adrenal axis, or HPA axis. Okay, and the amygdala also plays a role in activating the HPA axis, or the hypothalamic pituitary adrenal axis, and the hippocampus plays a role in inhibiting it. And like I said earlier, allostatic load happens when there's this core emotional area of the brain that's involved in responding to stress, and it's affected by the wear and the tear of the chronic stress. And this is the consequence of this cumulative overload and need to constantly adjust to psychological and environmental stress. Stressors are first detected by the brain's threat detection specialist, which is an interconnected network of sensory areas in the cortex, the thalamus, and the amygdala, like I said. And this is, the amygdala is involved in virtually all fear conditioning, and it works to jumpstart the stress-related networks peripheral to the central nervous system. You have two major peripheral systems subject to the central control of what's called the sympathetic nervous system. So the sympathetic nervous system, that's the fight or flight, and the parasympathetic is the rest, digest, and reproduction. You have this release of chemicals like epinephrine, adrenaline, norepinephrine or noradrenaline, cortisol, and those are the fight or flight hormones, okay? And they're going to divert the energy away from growth, from reproduction, from digestion, from all those things, okay? And you'll see these this arousal, the pupils change, the heart is racing, and then under normal conditions, like I said, that parasympathetic nervous system counteracts the sympathetic nervous system's effects, so it down-regulates the activity. Okay? Cortisol acts to increase the glucose levels in the bloodstream, and that's, you probably have heard a lot about the havoc that wreaks on your body after you've been exposed to stress for so long. Then we have overproduction of pro-inflammatory, what's called cytokines, that can occur, and so you can have a lot of different health issues and immune system issues when you're in chronic stress. Okay, so let's talk about the applications and we're done here. Early experiences do matter. Certain practices really do promote healthy prenatal development, and others put the fetus's well-being at substantial risk. Healthy baby guidelines promote healthy prenatal development, and those guidelines are, eating healthy, so fresh fruits and vegetables, avoiding alcohol and drugs. Fetal alcohol syndrome or fetal alcohol effects can happen at any time during the pregnancy. Okay, so it's important to completely avoid alcohol during pregnancy because we don't know when it can occur. Drugs, whether they're legal or illegal, find out what if it affects the fetus and what the effects are, whether it crosses the placental barrier. Cigarettes. There's a lot of things that happen with smoking, not just the lack of oxygen, but there's a lot of other issues. And we see babies who are drug exposed come out addicted. So reducing the stress is going to be another important guideline. Reducing interpersonal conflict as well. For some populations, primarily minorities, the stakes are even higher during pregnancy. The thought of this is disheartening and it's devastating. There's many psychosocial determinants of health, and we see that that microaggressions and the overt racism that is experienced by oppressed groups impacts their cortisol levels, and also the health disparities, healthcare disparities, access to care, things like that. Unfortunately. So prevention seems to be the key to reducing health disparities, and ultimately the number and the timing of exposure of stress during development is important for the kinds of disorders that chronic stress can bring about. I think that really we need to focus more on caring for our females from birth and beyond, even before they reach pregnancy. A lot of times our focus is only during pregnancy, but there's so much that leads up to that time when they're pregnant that's going to impact the fetal development.